So today's topic is minor carbohydrate metabolism. Please bear with me. Today I forget my laptop, so there is no PPT. Okay, just concentrate this board. I will try to explain you. Okay, so this is a very important topic. Minor carbohydrate metabolism. What is the major carbohydrate? Anyone? What is the major carbohydrate? No one. Glucose, very good. So glucose is abundant in our body. Every time we need glucose. Apart from that, there is some minor carbohydrate metabolism which actually does not require every time in our body. So in some disease condition, this carbohydrate poses some problem. Okay. Minor carbohydrate, what? Galactose, fructose, sorbitol. These things are very important. Okay. Lactose, we already discussed several times before. Lactose consists of what? It is a disaccharide. Please silence. Please silence. Okay, lactose is disaccharide. It consists of glucose and galactose. How it is metabolized in our body? চেয়ারটা সরিয়ে দিন না, শব্দ হচ্ছে বারে বারে। Galactose is metabolized by enzyme. Lactase. Lactose is metabolized by enzyme. Lactase. 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 Okay. In intestine, brush border, the lactase enzyme is there. Lactase broken down lactose into glucose and galactose. Okay, and then they absorb inside our body through the cell. Okay, in case of lactase deficiency, this enzyme deficiency causes some problem, like the lactose gets deposited in the intestinal lumen, and then bacteria causes a breakdown of the lactose in carbon dioxide, lactic acid, and causes some bloating, abdominal distension, diarrhea. Okay. So you may ask in your exam in lactose intolerance. This is called lactose intolerance. Okay. So this is one clinical scenario, lactose intolerance. Lactose intolerance. Okay, lactose intolerance. Yes, sir. So the question may ask you like this: one person after drinking milk or milk product, they experience bloating, abdominal distension, diarrhea, flatulence like this. What is the diagnosis? lactose intolerance. Very easy. Okay. So we have discussed this several times before. Okay. Now focus these three things. Galactose. What is galactose? It is a monosaccharide, right? And it is functional group is aldehyde. Very good. It is a epimerase of glucose. Okay. So galactose is a monosaccharide, functional group is aldehyde, and it is the epimer of glucose. It is also called as brain sugar. Okay? Why it is called brain sugar? Because in brain, galactose can form glycolipid present in nerve tissue. Okay? That is why it is also called as brain sugar. Okay? One type of glycolipid is formed in brain, and a glycolipid. Okay, so it present in nerve. It is also called as brain sugar. Now come metabolism. So how galactose is being absorbed in our intestine? It is absorbed by SGLT1. Okay, so this is galactose. It SGLT1. What is SGLT1? Sodium glucose transporter. SGLT1. It enters into the cell. Then it exists by GLUT2, GLUT2, glucose transporter 2, okay, and come here in bloodstream. Cytometabolism is mainly liver. Okay, cytometabolism mainly liver, galactose. One thing I have to like to mention, not only galactose, but glucose and fructose, everything transported by SGLT1. Okay. So how the metabolism occurs? It start galactose with galactokinase enzyme. It form one phosphate, galactose 1-phosphate. This is an irreversible reaction and it requires ATP. ADP. Okay. This is the enzyme galactokinase. Then next step is galactose 1-phosphate convert into with help of ADP glucose CO glucose UDP galactose. ADP glucose can convert into glucose 1-phosphate. Understand? Galactose convert into EDP galactose with the help of EDP glucose. What is EDP? Uridyl diphosphate glucose. Okay, and itself EDP glucose itself convert into glucose 1-phosphate. Okay, and it form EDP galactose. Enzyme is here. Okay, understood? Galactose 1-phosphate uridyl transferase enzyme. Understand? So, this is also another irreversible reaction. Galactose 1-phosphate convert into EDP galactose. Galactose. And with help of EDP glucose glucose itself convert into glucose 1-phosphate and it forms EDP galactose. Understand? Till now understand everyone? No question? কি বুঝতে পারছো না কিছু? Enzyme is galactose 1-phosphate uridyl transferase. Why you are talking, man? Please silent, na. Listen here. This is very important class, na. Why you are talking? Please listen. Okay. So galactose 1-phosphate convert into EDP galactose with the help of EDP glucose, and EDP glucose itself convert into glucose 1-phosphate. Okay. Now, what is the fate of this thing? EDP galactose and EDP glucose. EDP glucose convert into, glucose 1-phosphate convert into glucose 6-phosphate with the help of enzyme phosphohexose mutase. This is a reversible reaction. Okay. So glucose 1-phosphate convert into glucose 6-phosphate with the help of enzyme phosphohexose mutase. Phosphohexose mutase. Mutase enzyme. Just remember mutase enzyme. Okay. Next, what is the fate of this thing? Glucose 6-phosphate. বুঝতে পারছো না? আমার কথা মন দিয়ে শুনো, তাহলে বুঝতে পারবে। হ্যাঁ, glucose 1-phosphate convert into glucose 6-phosphate with the help of enzyme phosphohexose mutase enzyme. Understand? This is a reversible step, reversible reaction. Now, what is the fate of this thing? Glucose 6-phosphate. It will either go to glucose formation by the enzyme what? Or it may go into glycolysis. Understand? Glucose 6-phosphate either go into formation of glucose with the help of enzyme glucose 6-phosphatase It is present in endoplasmic reticulum. Okay. What the disease causes? Is the enzyme is deficient or defective? What is the disease? You have completed glycogen storage disease? Very good. Who said? Very good. Understand? Any question? Why are you smiling? Tell me why. You understand, na? Okay. This is present in endoplasmic reticulum. If the enzyme defect, it is called von Willebrand disease. Okay. What are the symptoms of von Willebrand disease? Hepatomegaly, fasting hypoglycemia, jaundice. Okay. So this is depend on if in fasting condition we require the glucose, so it convert into glucose. Okay. If you require energy, then it go into glycolysis pathway. Very simple. Okay. Now what is the effect of this thing? EDB glucose and EDB galactose. These things can interconvert. By enzyme 4' epimerase enzyme. EDB galactose can convert into EDB glucose. EDB glucose can convert into galactose by enzyme 4' epimerase enzyme. This is important to form of this thing form of lactose in mammary gland. So this enzyme is present in mammary gland during lactating mother. This enzyme is active. They convert galactose with glucose from lactose. Understand? 4' epimerase enzyme. Understand? Okay. And another thing is that this glucose 1-phosphate can convert into this thing, EDP glucose, by the enzyme EDP glucose pyrophosphorylase. Understand? Now listen carefully. Now this is come. What happened? Some disease condition. So this is one important enzyme. This is one important enzyme. If this enzyme is deficient, okay, if galactokinase enzyme is deficient, then one disease condition, it is called non-classical galactosemia. Okay, understand? If this enzyme is deficient, then one disease condition called non-classical galactosemia. What happens in this condition? So these galactose will accumulate in blood. Okay, so if this extra amount of galactose can convert into galactitol. This extra amount of galactose is combined into galactitol. Galactitol by the enzyme aldose reductase. Understand? Okay, and this is a troublesome thing. It accumulates in lens, eye lenses. It attracts water because this is osmotically active. Okay, and eye lenses lost the transparency. Causes one disease called what? Cataract. Okay, so in non-classical galactosemia patient, they develop bilateral cataract. Okay. Bilateral cataract. Apart from that, you found high level of galactose in blood, that is galactosemia, and escape through urine, galactouria. Okay, so you may ask in an exam, say 1 or 2 month old baby come to the OPD with mother complaining of haziness or whitish discoloration of both eyes, that is cataract. Apart from that, baby cannot see anything, he lost his sight. Okay, but apart from there is no any other symptom. So if you go for test, so galactose, he found that in urine it is Benedict test positive, because galactose is what? Reduced in sugar. Okay, and sorry. And if you go for blood test to see total galactose level by the process called ELISA, E-L-I-S, ELISA. You heard the name ELISA? He found high level of galactose. By this way you can diagnose. Understand? Now come very important, the second one. Okay. Second one, if this enzyme that is called galactose 1-phosphate uridyl transferase, in short form also called as GALT, G-A-L-T, GALT enzyme defect, this causes classical galactosemia. Okay, this is very, very important. You may ask in your exam classical galactosemia. Even in your life, when you start practicing, you may found some patients suffering from classical galactosemia. Okay, what are the symptoms of classical galactosemia? First of all, this enzyme defect. So what happened? Galactose 1-phosphate will start accumulate in the blood. Okay, you know, galactose combined to galactose 1-phosphate by galactokinase. So this enzyme defective, so you cannot convert further. So by the meantime, galactose phosphate start accumulate in the blood, and this causes a lot of problem. So what happened? The clinical symptom, this is bilateral cataract, one thing. Second, you found mental retardation. Three, you found jaundice. 4, you found fasting hypoglycemia. Okay, so classical galactosemia, you found these 4 symptoms very important. First one is bilateral cataract, next one is mental retardation, third one is jaundice, and fourth one is fasting hypoglycemia. Sorry, fasting hypoglycemia. Okay, so this enzyme defect causes this problem. Now, listen something. This GALT enzyme, this is enzyme, right? it it is a disease called autosomal recessive disease. What is autosomal recessive disease? There is a both allele are defective. Understand? Autosomal recessive disease. Next one is the gene located is called GALT gene, GALT, GALT gene defect. Gene is located 9p13. 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 These genes located in 9p13 chromosome. 9p13 chromosome. Okay? 9p13 chromosome. 9p13 chromosome. This is autosomal recessive disease. Incidence is 1:40,000 to 60,000 birth. Okay, incidence is 1 in 40,000 to 60,000 birth. Okay. Now we are discussing this thing. Why this biolactose? As I told earlier, because of deposition of what? Galactitol. Okay, because this enzyme defective, eventually galactose start accumulating. This galactose convert into galactitol by the enzyme aldose reductase. Okay. Next one is fasting hypoglycemia. So, why this fasting hypoglycemia? Because this galactose 1 phosphate galactose 1 phosphate, it inhibits glycogen synthase enzyme. Okay, glycogen synthase enzyme required ATP to synthesis glycogen phosphorylase enzyme. Please correct. Inhibit glycogen phosphorylase enzyme. Glycogen phosphorylase enzyme requires ATP. Okay, here maximum ATP is trapped here. Inorganic ATP trapped here in galactose 1 phosphate. Eventually, there is a depletion of ATP in some extent. Okay, glycogen phosphorylase enzyme 6 ATP to convert glycogen into glucose. Okay, so there is one cause of fasting hypoglycemia. Another thing is that this galactose 1 phosphate, it also inhibits this enzyme. Phosphohexose mutase. Okay, this thing galactose 1 phosphate also inhibit phosphohexose mutase. As a result, what happened? This glucose 6 phosphate cannot convert into glucose 1 phosphate. Okay, so glucose 1 phosphate supply is decreased. Eventually, what happened? Glucose 1 phosphate cannot convert into EDP glucose. Okay, EDP glucose is required for glycogen synthesis. Understand? Any question? Okay, so there is deficiency of glucose 6 phosphate. So eventually glucose 6 phosphate cannot convert into EDP glucose, and EDP glucose is required for glycogen synthesis. Okay, so there is second cause of fasting hypoglycemia. There is less glucose forming from glycogen breakdown. There is less glycogen synthesis because of less supply of glucose 6 phosphate. Understand? So you may ask in the exam, in classical galactosemia there is hypoglycemia, fasting hypoglycemia. Justify. So these are answers. Understand? Please write down this question. Understand? Next one is jaundice. Why there is jaundice? Because galactose 1 phosphate inhibit formation of the conjugation of bilirubin, for unconjugated to conjugated bilirubin you require ETP glucuronic acid. Understand? So there is less conversion of unconjugated to conjugated bilirubin. As a result, there is hyperbilirubinemia in the form of unconjugated bilirubin. Then it forms jaundice. What is jaundice? Jaundice is yellow discoloration of sclera and mucous membrane. Understand? One phosphate inhibit conjugation of unconjugated bilirubin to conjugated bilirubin. As a result, there is hyperbilirubinemia in the form of unconjugated hyperbilirubinemia. Galactose 1 phosphate inhibit conjugation of unconjugated bilirubin to conjugated bilirubin. Okay. Jolly. So there is jaundice. And last one is mental retardation. Why there is mental retardation? Cause is still unknown. Probably, as I told earlier, galactose is a brain sugar. So there is less, I don't know why, probably actually this cause is still unknown. But still, because probably there is less ATPs there, because all the inorganic ATP is trapped here in the form of galactose 1 phosphate. So there is less ATP, so there is a start of neurodegeneration. Okay, it may lead to mental retardation. Okay, so everybody understand, na? Okay. This is called classical galactosemia. Next term, fructose. What is the metabolism? fructose combined to fructose 1 phosphate by the enzyme fructokinase. Fructose is next converted into fructose is first combined to fructose 1 phosphate by the enzyme fructokinase. Okay, here also we record ATP. Okay, next fructose 1 phosphate is split into dihydroxyacetone phosphate and glyceraldehyde. Understand? Fructose 1 phosphate is split into dihydroxyacetone phosphate and glyceraldehyde by the enzyme aldose B enzyme. Okay, aldose B enzyme. Okay, next this enzyme dihydroxyacetone phosphate is going to glycolysis pathway. Ultimately form pyruvate by the enzyme pyruvate kinase. And pyruvate kinase ultimately form acetyl-CoA by pyruvate dehydrogenase enzyme. Okay, glyceraldehyde, sorry?
It's not visible. The writing is quite small.
Quite small. Just listen to me. Aldose B split into dihydroxyacetone phosphate and glyceraldehyde. Understand? Glyceraldehyde go into glycolytic pathway. Ultimately form pyruvate by the enzyme pyruvate kinase. Pyruvate test form acetyl-CoA by pyruvate dehydrogenase enzyme. You understand? Okay. What is the fate of this glyceraldehyde? Glyceraldehyde combined to glyceraldehyde 3 phosphate, and again this into going to glycolytic pathway. Okay, and this thing can interconvert. You already learned in glycolytic lectures, na? Glycolysis complete, na? Tomorrow class glycolysis complete. Okay, this thing can interconvert by triose phosphate isomerase enzyme. Understand? So fructose is first converted into fructose 1 phosphate by the enzyme fructokinase with the help of ATP. This is a irreversible reaction, and then fructose 1 phosphate is split into dihydroxyacetone phosphate and glyceraldehyde. Okay? Dihydroxyacetone phosphate can interconvert into glyceraldehyde. Dihydroxyacetone phosphate go into glycolytic pathway, ultimately form pyruvate. Pyruvate can form acetyl-CoA Okay. Glyceraldehyde can convert into glyceraldehyde phosphate by triose enzyme. Okay. This thing can intercompare dihydroxyphosphate and glyceraldehyde phosphate already you taught in glycolitic lectures. They can interconvert by thiophosphate or phosphothiose isomerase enzyme. Okay. So this is the fructose metabolism. What is the difference between fructose metabolism and glucose metabolism? In glucose metabolism, there is formation of glucose to glucose 6 phosphate by enzyme hexokinase or glucokinase. Okay. Ultimately form fructose 1,6 bisphosphate. Here one important enzyme is there called phosphofructokinase PFK 1. Okay. In glycolysis, this is tightly regulated. Okay, but you see in fructose there is no such regulations there. Fructokinase can be regulated by either 2,3-dephosphoglycerate or less AMP. You can activate. You can inhibit by hydrogen ion, citrate, etc. So these things can regulate, but here it cannot regulate. So fructose is actually a keto sugar. It is monosaccharide. Okay, it present in cold drinks, fruit juice. Okay, so if you drink lot of cold drink there is lot of sugar, table sugar they use. Okay, so ultimately form lot of acetyl CoA. Acetyl CoA can convert into fatty acid. Okay, so if you drink lot of sugar, lot of cold drink, eventually you end up with high fatty acid content. It forms hypertriglyceridemia, obesity. Fatty acetyl CoA is required for cholesterol synthesis. There is hypercholesterolemia. So understand? So if you drink lot of cold drinks, eventually you end up with obesity. Understand? Now come important two enzyme, fructokinase and aldose B enzyme deficiency. Fructokinase deficiency is the disease called essential fructosuria. Essential fructosuria, this is a benign condition. There is no any other symptom present. Only when you do urine test, there is positive selenium test. তোমাদের বলা হয়নি ইউরিন ফ্রুক্টোজ সেলিওয়ান টেস্ট হ্যাঁ হয়নি কেন হয়ে যাবে জাস্ট রিমেম্বার ওকে সেলিওয়ান টেস্ট Sally one of please, my handwriting is very poor. test is positive in case of fructose. Okay, this is essential fructosuria. Okay. Next is this enzyme defect, aldose B defect, also called fructose 1-phosphate aldolase. Defect causes TA hereditary this enzyme defect cause hereditary fructose intolerance. Hereditary fructose intolerance. Understand? Hereditary fructose intolerance. This enzyme defect causes this disease. Okay. So what is the symptom? Symptom is like classical galactosemia, except there is no cataract. Okay. Just like classical galactosemia, there is no cataract. Because you know fructose is a keto sugar. So it cannot combine to fructitol. Understand? As the fructose is keto sugar, it cannot combine to fructitol. Just remember. So there is no cataract. Apart from that, there is all the other symptoms are just like galactosemia, like fasting hypoglycemia because of this thing. This fructose 1-phosphate is inhibit the conjugation of unconjugated bilirubin to conjugated bilirubin. So there is jaundice. Okay, it inhibit glycogen phosphorylase enzyme. So there is no formation of glucose from glycogen breakdown. So there is fasting hypoglycemia. Understand? And there is also mental irritation because of, as I told earlier, fructose 1-phosphate here, phosphate get trapped. Okay? The diagnosis is by urine test, positive selenium-1 test. Okay? Any question till now? Any question? Understand?
Sir, difference between classical and non-classical galactosemia?
Difference is only one thing. In classical galactosemia, there is mental retardation, there is jaundice, there is fasting hypoglycemia. Okay? In non-classical, there is only cataract.
क्लासिकल और नॉन क्लासिकल दोनों में बाइक
Okay. Last one is sorbitol. Okay, so sorbitol is also called as भूल गया मैं. So glucose is converted into sorbitol when there is much greater amount of glucose is there. Because this enzyme aldose reductase is actually high Km value. So it is less affinity towards glucose. Okay. So when there is high glucose, in case of uncontrolled diabetes, there extra amount of glucose can convert into sorbitol. Okay, and sorbitol causes a lot of problem. Again, this sorbitol convert into fructose by enzyme sorbitol dehydrogenase with use of NAD and NADH plus H+. Okay. Sorbitol is actually osmotically active. So in case of lens, it attract water, causes cataract. So in case of diabetes, there is cataract formation, accelerated cataract formation because of deposition of sorbitol. Okay. Again, this sorbitol converted into fructose. This particular enzyme is very important. Sorbitol dehydrogenase is present everywhere, especially in testes and seminal vesicle. It forms fructose because fructose is required for spermatogenesis. Understand? But in other organ where this enzyme is deficient, like lens, kidney, retina, next one is swan cell, SW, swan cell of nerve, it causes problem. It lens, it causes cataract because of deposition of this thing, sorbitol. And kidney, diabetes, it causes nephropathy. Retina causes retinopathy. And swan cell of nerve causes neuropathy. Okay. The deposition of sorbitol in lens causes cataract, in kidney causes diabetic nephropathy. This is one cause actually. There is a lot of other causes there, like glycosylation of protein causes nephropathy. This is one cause. There is several other causes there. This is one cause. And retina causes retinopathy. In swan cell of nerve causes neuropathy because sorbitol attract water and it damages these organs. Understand? Enzyme is aldose reductase convert glucose into sorbitol into fructose. Okay, aldose reductase also used where galactose from galactitol, that is also osmotically active. Understand? Swan cell of nerve. Swan cell of nerve causes neuropathy. Any question? So please go through your book today. This is a very important class for clinical perspective of view. Okay, please. Any problem? Sorry? Liver. Not liver. This is lens. Lens. Please come here. Sit forward. Bolo.
Test for fructose.
Saliva test. Thank you very much. Today class is over.